A pill looks simple. A capsule, a tablet, a vial. But behind every medicine lies one of the most complex, expensive, and regulated processes on Earth. A journey that can take more than 10 years and cost over $1 billion. The story begins with a question. A disease with no cure. A biological pathway that scientists barely understand. A molecule that might, just might, change everything. This first stage is discovery research. Scientists study cells, proteins, and genes to find a target. Something inside the body that can be modified to treat the disease. Sometimes the target is a receptor, sometimes an enzyme, sometimes a mutation. This is where biology meets imagination. Drug discovery. Once the target is found, chemists design thousands of molecules. They test each one in the lab. Most fail. A few show promise. One becomes the lead compound, the molecule that moves forward. Preclinical testing. Before a drug ever reaches a human, it must survive a stage that is both scientifically essential and ethically complex. Preclinical testing. This is the part of drug development that most people never see. A world of controlled environments, specialized facilities, and highly trained scientists. A world where the first question is not, does it work, but is it safe? Cell and tissue studies. The first filter. The molecule is tested in human cell lines, organoids, engineered tissues, biochemical assays. Researchers measure toxicity, metabolism, and how the compound interacts with biological pathways. But cells can only tell part of the story. To understand how a drug behaves inside a living organism with blood flow, organs, metabolism, and immune responses, scientists must use animal models. Why animals are used? Animals provide a whole-body system that cannot be replicated fully in vitro. They allow researchers to study absorption, distribution, metabolism, excretion, organ toxicity, long-term safety. These are the eudaimonia properties that determine whether a drug can survive inside a living organism. The species used, each with a purpose. Different animals answer different scientific questions. Mice, genetics, cancer, immunology, rapid testing. Rats, toxicology, metabolism, dosing studies. Rabbits, ocular and dermatological safety. Dogs, cardiovascular and pharmacokinetic studies. Pigs, organ physiology similar to humans. Non-human primates, used only when absolutely necessary for complex immune or neurological diseases. Each species provides unique biological insights. The choice is never random. It is dictated by regulatory guidelines and decades of comparative physiology research. The environment, controlled to the millimeter. Animal research facilities are highly regulated. Temperature, humidity, light cycles, air filtration, everything is controlled. Animals receive monitored diets, veterinary care, enrichment to reduce stress, controlled handling procedures. Every experiment follows strict ethical frameworks such as the 3 Rs. Replace animals when possible. Reduce the number used. Refine procedures to minimize suffering. Preclinical testing includes 1. Acute toxicity studies. Single high doses to identify dangerous effects. 2. Chronic toxicity studies. Repeated dosing over weeks or months to detect long-term risks. 3. Pharmacokinetics. How the drug moves through the body. Absorption, distribution, metabolism, excretion. 4. Pharmacodynamics. What the drug does to the body, effects on organs, tissues, and biological pathways. 5. Safety pharmacology. Impact on heart, lungs, brain, liver, kidneys. 6. Reproductive and developmental studies. Effects on fertility, pregnancy, and offspring. 7. Carcinogenicity studies. Long-term risk of cancer. Every result is recorded, analyzed, and compared to strict regulatory thresholds. If the drug shows acceptable toxicity, predictable metabolism, manageable side effects, and therapeutic potential, it becomes a drug candidate. If not, the project ends, sometimes after years of work. Clinical trials, the human journey. Once regulators approve the data, human testing begins. Phase 1, safety first. A small group of healthy volunteers. Researchers study side effects, dosage, and how the drug behaves inside the human body. Phase 2, does it work? Patients who actually have the disease. Scientists measure improvements, biomarkers, and compare results to existing treatments. Phase 3, the global test. Thousands of patients across multiple countries. Hospitals, clinics, laboratories, all synchronized. This phase determines everything: effectiveness, safety, long-term impact, and comparison to standard therapies. Many drugs fail here. Manufacturing, engineering, and medicine. Factories produce tablets, capsules, or injectable solutions with extreme precision. Temperature, humidity, purity, everything is controlled. Machines test each batch. Quality control is relentless. Distribution, the global supply chain. The drug travels through warehouses, hospitals, pharmacies, and cold chain logistics. Tracking systems monitor every shipment. The medicine reaches patients in Madrid, New York, Tokyo, Nairobi. Post-marketing, the final phase. After approval, the drug enters Phase 4 monitoring. Real-world surveillance. Doctors report side effects. Scientists study long-term outcomes. Regulators update guidelines. The drug continues to evolve as new data emerges.